water-soluble molecules that play a crucial role in human metabolism. These specialized compounds contain ketone groups and serve as an alternative energy source when our body's primary fuel, glucose, becomes limited. These molecules are characterized by their ketone functional groups, which consist of a carbon atom double bonded to an oxygen atom. This chemical structure makes them water-soluble, allowing them to travel easily through our bloodstream. There are three primary ketone bodies that our body produces. First is acetoacetate, the initial ketone body formed. Second is beta-hydroxybutyrate, which is actually the most abundant ketone body in our blood. Third is acetone, which is produced in smaller amounts and is often exhaled through our lungs. Ketone bodies are primarily produced in the liver through a process called ketogenesis. When fatty acids are broken down in liver cells, they can be converted into these ketone bodies. This process becomes especially important when glucose availability is low. Ketone bodies serve as a crucial alternative fuel source for our body's most energy-demanding organs. The brain, heart, and muscles can all efficiently use ketone bodies for energy when glucose becomes scarce. This metabolic flexibility is essential for survival during periods of fasting or low carbohydrate intake. One of the most important properties of ketone bodies is their ability to cross the blood-brain barrier. This protective barrier normally prevents many substances from entering the brain, including fatty acids. However, ketone bodies can pass through this barrier, making them uniquely valuable as brain fuel when glucose is unavailable. This diagram illustrates the complex metabolic interactions between liver cells, the bloodstream, and brain cells. We can see how fatty acids are processed in the liver and how various metabolites, including ketone bodies, are transported through the blood to provide energy for brain function. When ketone body production exceeds their utilization by tissues, blood ketone levels rise, creating a metabolic state called ketosis. This is a natural and often beneficial adaptation that allows the body to efficiently use fat for energy when carbohydrates are limited. Understanding ketone bodies is fundamental to appreciating how our metabolism adapts to different nutritional states. These remarkable molecules demonstrate the body's incredible ability to maintain energy supply to vital organs, particularly the brain, even when our primary fuel source becomes scarce. Ketogenesis is the biochemical process where the body produces ketone bodies from fatty acids. This vital metabolic pathway occurs primarily in the liver and serves as an alternative energy production system. Ketogenesis takes place mainly in the mitochondria of liver cells, called hepatocytes. These specialized cells contain numerous mitochondria where the ketone body formation process occurs. Ketogenesis is triggered when blood glucose levels are low. This occurs during fasting, starvation, following low-carbohydrate diets, or during intense exercise when the body needs alternative fuel sources. The process begins with beta oxidation, where fatty acids are broken down in the mitochondria to produce acetyl-CoA molecules. This is the first step that provides the building blocks for ketone body formation. When oxaloacetate becomes depleted, often because it's being used for glucose production, acetyl-CoA is redirected into the ketogenesis pathway. This is a crucial metabolic switch that allows the body to produce alternative fuel. The pathway begins when two acetyl-CoA molecules combine to form acetoacetyl-CoA. This is then converted to HMG-CoA by the enzyme HMG-CoA synthase, followed by cleavage to acetoacetate by HMG-CoA lyase. From acetoacetate, the body produces three different ketone bodies. Acetoacetate can be reduced to beta-hydroxybutyrate, or it can spontaneously break down to form acetone, which is often exhaled through the lungs. Ketogenesis is a sophisticated metabolic adaptation that allows the liver to convert fatty acids into water-soluble ketone bodies. This process ensures the body has alternative fuel sources when glucose is limited, particularly benefiting organs like the brain that have high energy demands. Ketolysis is the process by which tissues throughout the body utilize ketone bodies as an alternative energy source. This metabolic pathway allows organs to efficiently convert ketone bodies back into usable energy when glucose is limited. Ketone bodies are transported from the liver to other tissues via the bloodstream. Unlike fatty acids, ketone bodies are water-soluble, making them easily transportable through the circulatory system. Importantly, the liver cannot use ketone bodies for energy because it lacks the enzyme thiophorease, also known as beta-ketoacyl-CoA transferase. This ensures that ketone bodies produced by the liver are available for other tissues that need them. Now let's examine the biochemical pathway of ketolysis. This process involves several key enzymatic steps that convert ketone bodies back into acetyl-CoA, which can then enter the citric acid cycle for energy production. The first key step involves the enzyme SCOT, or succinyl-CoA colon 3-ketoacid-CoA transferase. This enzyme converts acetoacetate into acetoacetyl-CoA, preparing it for the next stage of the pathway. The second crucial step involves acetoacetyl-CoA thiolase. This enzyme cleaves acetoacetyl-CoA into two molecules of acetyl-CoA, effectively doubling the number of molecules that can enter the citric acid cycle. Finally, the acetyl-CoA molecules enter the citric acid cycle, also known as the TCA cycle or Krebs cycle. Through oxidative phosphorylation, this process generates ATP, the universal energy currency of cells. Ketolysis is a vital metabolic pathway that allows tissues throughout the body to efficiently utilize ketone bodies as an alternative energy source. This process ensures that when glucose is limited, organs like the brain, heart, and muscles can continue to function by converting ketone bodies back into the acetyl-CoA needed for energy production. Brown adipose tissue, commonly known as BAT, plays a crucial role in recycling ketone bodies within our metabolic system. Brown adipose tissue is a specialized type of fat tissue that is metabolically very active. Unlike regular fat tissue, BAT consumes excess lipids and converts them into ketone bodies. The enzyme acetoacetyl-CoA synthetase, or AACS, is the key player in ketone body recycling. This enzyme converts ketone bodies back into building blocks that can be used for lipogenesis, the creation of new lipids. This recycling process is essential for managing energy balance within the body. By converting ketone bodies back to lipids when needed, the body maintains metabolic flexibility and ensures efficient energy utilization. The complete recycling cycle shows how brown adipose tissue produces ketone bodies, which are then recycled by the AACS enzyme back into lipids. This creates a continuous cycle that maintains metabolic balance and ensures the body can efficiently manage its energy resources. This recycling mechanism demonstrates the sophisticated ways our body manages energy metabolism, ensuring that ketone bodies are not just used for immediate energy needs, but can also be recycled back into storage forms when appropriate. Ketone bodies are primarily produced in the liver mitochondria, which serve as the body's main ketone manufacturing center. However, they can also be produced in smaller amounts by glial cells in the brain and kidney epithelial cells. Ketone bodies are water-soluble molecules, which makes them easily transportable through the bloodstream. Unlike fatty acids, they don't require special carriers and can move freely in the aqueous blood environment to reach tissues throughout the body. One of the most important features of ketone bodies is their ability to cross the blood-brain barrier. They use specialized transport proteins called monocarboxylate transporters, or MCTs, to enter the brain tissue where they can fuel brain cells. Once ketone bodies reach their target tissues, they serve different roles. In the brain, they become a primary energy source when glucose is limited. In the heart and muscles, they provide additional fuel to support cellular energy demands. The energy production from ketone bodies occurs through their oxidation in cellular mitochondria. This process yields ATP, NADH, and FADH2, which are essential molecules that drive the electron transport chain to produce the energy cells need to function. Ketone bodies play crucial roles in two important metabolic scenarios: during starvation, when the body runs out of stored carbohydrates, and during ketogenic diets, when carbohydrate intake is deliberately restricted. During starvation, the body goes through distinct phases. Initially, it uses stored glycogen for energy, but these stores are limited and become depleted within 12 to 24 hours. Once glycogen stores are exhausted, the body shifts to breaking down fat for energy. This process dramatically increases ketone body production as an alternative fuel source. However, prolonged starvation can be dangerous. When ketone production becomes excessive, it can lead to starvation ketoacidosis, particularly in individuals with poor dietary intake or or underlying health conditions. Ketogenic diets are high-fat, low-carbohydrate diets that deliberately induce ketosis. While a normal diet contains about 50% carbohydrates, ketogenic diets restrict carbs to just 5% of total calories, with 75% coming from fats. By restricting carbohydrate intake, ketogenic diets shift the body's primary fuel source from glucose to fat. This metabolic switch increases ketone body production, creating a state called nutritional ketosis. Nutritional ketosis is generally considered safe for healthy individuals. The ketone levels typically range from 0.5 to 5 millimolar, which is well below the dangerous levels seen in ketoacidosis. This controlled state of ketosis provides an efficient alternative energy source while maintaining metabolic safety. Recent research has revealed that ketone bodies serve roles far beyond simple energy production. They act as powerful signaling molecules that influence cellular processes, protein quality control, and aging mechanisms throughout the body. The three main ketone bodies, acetone, acetoacetate, and beta-hydroxybutyrate, each have unique signaling properties. Beta-hydroxybutyrate, in particular, has emerged as a key signaling metabolite with profound effects on cellular